Hi, my name is Marta Wayne, and I am the past president of SMBE. I am honored to have been able to serve the Society for the last few years, and also very grateful to be able to give the second SMBE presidential address this year, as due to the cancellation of our meeting due to COVID, I was unable to do it last year. I'm going to talk with you today about neutral theories, past and present. I'll start with the neutral theory of molecular evolution, which was created by Moto Kimura. According to this theory, most new mutations are quickly lost due to chance, including beneficial mutations, unless they have a very large selective advantage. And of course, most new mutations are unconditionally deleterious. However, a mutation may be considered effectively neutral if its selective coefficient is small relative to the effective population size, NE. Neutral mutations fix at a rate of 1 mutation per every 4 NE generations. The neutral mutation rate equals the rate of neutral evolution, that is, the neutral substitution rate. Nu, the neutral mutation rate, equals K, our evolution rate. Here is a picture from Kimura's 1983 book depicting how the neutral theory of molecular evolution works. If you look at the x-axis, this is time in terms of the number of generations, and the y-axis is frequency, running from 0 to 1. When we talk about a mutation fixing, that means it rises to a frequency of 100%. The heavy lines represent neutral mutations that are destined to fix. And you can see that this neutral mutation fixed, and it took 4 NE generations to do that. You could also see that a neutral mutation will fix proportional to the rate of Nu, the neutral mutation rate. What you can't see on this picture is that there is a constant input of strongly deleterious mutations, which are instantly eliminated, and also neutral mutations, most of which are lost due to chance. We also have a few neutral mutations that at any given snapshot in time we could see, but they don't fix. When I began graduate school in 1988, the neutral theory was still pretty exciting and pretty new. After a brief bout of studying speciation genetics unsuccessfully, I switched over to working on what the rest of the lab was doing and began to study linkage and selection. The idea here is that linkage to a selected site will temporarily lead to reduced neutral variation around that site, a selective sweep. The reason for this is that the selected site fixes very quickly, and all of the neutral variants that are linked to it will also fix. So that results in an appearance of no variation around the site. I'm going to show you how this would work using Kimura's figure. So we're going to have a strongly advantageous mutation arise here. That mutation will fix much faster than the neutral mutations, and it is arising in linkage disequilibrium, that is, on the same chromosome as these existing neutral mutations. So they're going to fix at the same time, and that means that they're going to disappear. So here you go. Our strongly selected mutation has fixed, and the result of that is that the neutral variation in the region is now gone. So variation will of course be recreated via neutral mutation, Nu, and then our selective sweep will gradually disappear. It will disappear at a rate proportional to the rate of recombination. And the reason for this is that the neutral variants are going to appear on different chromosomes, and so they'll be in linkage disequilibrium with our selected allele, but eventually recombination will homogenize things, and that window of reduced variation, the sweep, will be gone. Therefore, regions of reduced recombination should show less variation within species relative to regions of high recombination. However, if recombination itself were mutagenic, then we would see the same positive association between neutral variation within species and recombination, as well as an observation that neutral divergence between species should also increase with recombination. In 1992, Begun and Quadro collected variation data from Drosophila melanogaster and tested this theory. They showed that recombination on the x-axis, the coefficient of exchange, was indeed positively correlated with nucleotide diversity, pi, or neutral variation within species, on the y-axis. They also did the critical experiment to see whether or not this relationship held across species as well as within species. And what they found was that there was no relationship between variation among species and the coefficient of exchange. This matters because it showed that recombination was not obviously mutagenic per se and could not explain the relationship observed within species. This is really exciting and one of the early tests of the neutral theory. Now, my work added 2 points to these figures, both of which fit nicely with the previous observations. There were a number of other aspects to the work that I did for my dissertation, which I will return to later, at least some of them. Now, in addition to my fascination with speciation genetics, I also felt the dark call of dosage compensation. Anyone who works with Drosophila will recognize this beautiful chromosome squash. I credit the Max Scott lab from NC State for this lovely image. And this chromosome here is the X chromosome in a male fly, which is being upregulated to dosage compensate for the 2 X chromosomes in a female. In other words, by upregulating the X in males, flies accomplish dosage compensation, making expression in the male X equivalent to the female X and to the autosomes. I began to wonder what a neutral theory for the evolution of dosage compensation might look like. I worked with David Hall, and we started from the simple point that sex chromosomes arise from autosomes, which have a substantial load of recessive hypomorphic mutations given mutation selection balance. It's worth spending a moment to explain what a hypomorphic mutation is. So a hypomorphic mutation is a reduction in function mutation. And in this context, that could be because a gene is expressed at a lower level than wild type. It could also be that the gene product is less functional than a wild type. So not a knockout, just with lower efficiency. And once a male determining locus arises, alleles conferring male benefits should accumulate in linkage disequilibrium with the sex determining locus, and selection pressure for reduced recombination will arise. I've put an asterisk by male here because this works just as well for female heterogametic sexes as well as for male heterogametic sexes. I've just used male as a shorthand here because I'm a Drosophilist. Now, once that selection pressure for reduced recombination arises, because we want to maintain the linkage disequilibrium between the alleles conferring male benefits and the male determining locus, then we're going to see that genes are going to degrade in the non-recombining regions of the neo-Y or the neo-W, and that will result in hemizygosity. Hemizygosity meaning a single copy of a gene where once there were 2. Now, a cost of hemizygosity can either result from hemizygosity per se, as expressed in Ono's peril of hemizygosity. That is to say that genes should be expressed in an equilibrium between sex chromosomes and autosomes, such that genes on a male chromosome should be expressed at the same rate as genes on a female X or autosomes of males. However, a cost of hemizygosity can also arise from unmasking segregating hypomorphic alleles. These alleles are not common, but they are segregating at some frequency on autosomes. And when they appear opposite a null or in the state of hemizygosity, then they are subject to selection. We considered that ideal compensation would match the average diploid expression state, i.e. twice the homozygous expression level of a wild-type allele. we included a possibility of a fitness cost of overcompensating, that is, creating hypermorphic alleles, as well as the cost of undercompensating. So when an allele is hemizygous, it is not expressed at the same amount as it would be if it were a diploid allele. And we can imagine that if the expression level of our wild-type diploid allele is optimum, there could also be a fitness cost of overexpressing that allele. And so we included this in our model. Finally, we linked expression with fitness by using the metabolic control theory framework of Ketcher and Burns, 1981. So we're going to spend a couple of minutes talking about how that works. The metabolic control theory proposed a molecular mechanism for dominance, somewhat akin to what Wright was thinking of in the 30s. It's a slightly complicated figure, so let's walk through it slowly. The x-axis is E, the enzymatic activity of a focal enzyme, with 1 being wild type, 0 being a complete knockout. The y-axis is the flux through the pathway. Again, let's have 1 be the wild type flux, 0 be no flux. In other words, when the enzyme is working correctly, we have a flux of 1. So if our pathway consists of a single enzyme, obviously we're going to see a linear relationship between the function of that enzyme and flux. However, the more enzymes we add to the pathway, the more concave and nonlinear this function becomes. So with 5 enzymes, a change in the activity of our one enzyme has less of an effect. And if we have 20 enzymes, then a change in the efficiency or the activity of our single enzyme has an even smaller effect on total flux than you might expect from a single enzyme pathway. What does this all mean with respect to dominance? Well, recall that dominance is a deviation from additivity. So let's imagine that we have an individual who is heterozygous for a wild-type allele with complete enzymatic activity and a knockout allele with 0. Under additivity, we would expect to see that the enzyme efficiency is half that of wild-type, and we would also see that the flux through the pathway is also 50%. But if you imagine instead that we're looking at a heterozygote between a wild type and a null for one enzyme and a pathway of 5, we see something different. We see that instead of our heterozygote resulting in 50% flux, it results in slightly more than 80% flux. And if we use instead a pathway of 20 enzymes, we're actually getting greater than 95% flux through the pathway, even though we have a heterozygote with a null. This is the manifestation of dominance. In order to do our study, we did a little sleight of hand and explicitly equated flux through the pathway with fitness, such that wild-type flux of 1 is fitness of 1. Without going through the beautiful mathematics and simulations, we'll talk instead about the results for the sake of time. So either hemizygosity or hypomorphic alleles will drive increased expression, i.e. dosage compensation, under our model. Unmasking of hypomorphic alleles is more important for increasing expression earlier in sex chromosomes evolution than later. And because the loss of genes in longer pathways has little cost, these genes will be lost earlier in the evolution of a new sex chromosome than those of shorter pathways. And in fact, the loss of genes in short pathways is unlikely to occur until dosage compensation has fully evolved. I mentioned that I would get back to some aspects of my dissertation research later on. Here you go. This is a slide of rhabdoviruses amidst some cellular debris. One of the genes I studied was a resistance gene for a virus in Drosophila melanogaster, sigma virus, which is also a rhabdovirus. Through a variety of experimental evolution studies in this system, which I don't have time to discuss now, I became interested in whether or not we could also have a neutral model for the evolution of virulence. So let's talk for a moment about virulence evolution. Specifically, I'm going to define virulence as a reduction in host fitness caused by an infection, a parasite, a pathogen. And we expect virulence to be evolvable because it involves both the genome of the parasite causing the infection and the genome of the host. Currently, the most popular theory for the evolution of virulence is the intermediate optimum, or trade-off theory. There are two assumptions of this theory. The first is that increased within-host replication rate increases transmission. Transmission is effectively viral fitness or parasite fitness. So transmission to new hosts means higher fitness. Increased within-host replication also increases the probability of host death, which then would decrease transmission, because dead and dying hosts, generally speaking, encounter fewer new hosts than less sick individuals would. So here's a visualization of the intermediate optimum theory. The idea then is that we've got virulence on the x-axis and we've got parasite fitness on the y. As virulence increases and transmission goes up, for a while fitness is also going up. But at some point, as virulence becomes too great and our host begins to become too sick to move around and socialize with other hosts and or even dies, then parasite fitness drops off because new hosts are not being infected. This gives rise to an intermediate optimum for virulence. So I wondered about what a neutral model for the evolution of virulence might be and began collaborating with Ben Bolker and his graduate student Morgan Kane probably around 2017. It's not entirely clear when. And this was really exciting. We talked about the different ways that we could reframe these assumptions, the role of deleterious mutations, and so forth. And then things started to happen. So as the chair of a large biology department at a public institution in the US Southeast, I began to focus my energy more on these issues and had to walk away from this project for a time. You can ask Ben and Morgan a little bit about the progress of this now. You can ask me about it later when I have a chance to return to it. For now, however, I'm going to talk to you about something very different, and that is to think about a neutral model for participation in science. Our starting assumption then is that talent is equally distributed among geographic regions, races, genders, ethnicities, socioeconomic classes, abilities, and so on. If that is true, then our neutral expectation would be that participation in science should reflect the international diversity of humanity. That is, any given group should be represented in science proportional to their representation in the global human population. I don't think I need to show you data for you to believe that we have falsified that neutral expectation. Given that that's the case then, what is the current model for participation in science? How can we think of this? Before I talk further about this, I want to talk about the qualities that allow people to persist in science, the qualities that allowed me to be sitting here today giving this address to you, the qualities that are allowing you to listen to this address. I'm often asked to describe the qualities of successful scientists, either informally or as part of mentoring programs with students from a wide range of levels, from primary school on up to my fellow faculty. In the past, these are the three adjectives I have used: stubbornness, resilience, and creativity. The reason I use these adjectives, and you will notice that intelligence, aptitude, talent is not listed, is because ours is a field of continuing rejection. It is a field where you are getting your papers rejected. You may get rejected from university. You get rejected from job applications. You get rejected from grant proposals. You even get rejected sometimes from society elections. So you're getting rejected all the time. In order to persist, you have to be resilient. You have to be able to pull yourself back up, and you need to be creative. You may need to reframe your science in order to succeed. When I give this talk the next time, I'm going to include another adjective, and that adjective is access. Because in addition to all of these things, in order to succeed in science, you need to have access. You need to have had access to appropriate education and training. You need to have access to a community and mentors who not only accept you, but affirm you and help you to succeed. So let's return to this question of what is the model for participation in science? This is a challenging question, and one model would be the meritocracy, the idea that the best are the people who are still here. And that's a nice idea. It's nice to consider oneself the best. But if I look at myself, I see a very good scientist. I see an excellent scientist. I do not see a scientist who is in the top 100th of the top 1 percentile. But I'm still here. And the question then is, why? I think that a better explanation for successful participation in science is probably the patronage model. The idea being that if one has a patron, one is more likely to succeed. Again, to use myself as an example, I had the wonderful fortune of joining the lab of Marty Kreitman as a graduate student. And what that meant is that people took me seriously and were willing to listen to me long before I had earned the right because they knew Marty's name. Marty was also a fantastic mentor. And one of the things that he did that was so wonderful is that he viewed me and treated me as a colleague. In fact, Marty did not recognize me as a woman in science until I was an assistant professor when we had a very humorous phone call and he said, "Wow, wait, you're a woman." I was like, dude, that's true. The reason this matters is because being treated like a colleague and not as a something else is the most important gift that you can give your mentees. It's the most important gift that I received. Another example, though, about patronage would be the efforts and promotions that Bill Martin gave me. He helped me be an associate editor. He helped me learn how to do this, and he did it because I was a woman. He was addressing the problem of gender equity on the editorial boards. And this was also a wonderful gift, but a very different one. These are two examples of patronage, and I don't think the patronage system is a good system. It limits participation by definition. I'm giving these examples to show you how things can change and to help you think about what the appropriate model for participation in science should be. I will also argue that a neutral model isn't going to work because we don't have neutral processes operating in determining who can and cannot participate in science. I'm now going to move on and show a figure about equity in science. So I'm going to talk a little bit about justice in science and in SMBE. And if you are in the US, you have probably seen endless variations on this figure this year. It's contrasting equality and equity and justice. And the usual figure is a bunch of people trying to see a baseball game over a fence. I chose this version, which was developed by Avarna, which is an organization working to promote equity and provide resources for those seeking equity in environmental sciences, conservation, and outdoor leadership activities. I like it to begin with because it's less culturally biased. We've got people trying to watch the sun, watching a sunset. So an important thing to notice here is that all of these figures are the same height. That's important because it's not implying that there's a group that started with a deficit. We could also critique this because all the figures look exactly the same, and of course we're not all exactly the same. But leaving that aside for the moment, we should also notice that each one of them has a box. That's equality. Everyone has the same resources. But because they are not standing on a level surface, everybody can't see over the fence. Again, to borrow from my experience, starting out in graduate school, I would have put myself in the middle position. So advantaged because I am born in the US to a white middle-class family. I had access to great schools. My parents respected science. They liked learning. They could understand that a PhD was almost as good as an MD, and I even convinced them that pursuing a career in academia was a rational choice. That was a lot of support. That's a lot of access. Now our next panel shows equity. Equity means that we have redistributed the resources, the boxes, so now everyone can see over the fence, even though there's a non-level surface. So we've created custom interventions that help unique people, unique groups be more successful. And this is great. But it's not justice. Justice is when there's no fence. If there were no fence, no one would need boxes to see over that fence and watch the sunset. And sunsetting into the ocean, this is a beautiful thing. But there is a panel, in my opinion, that is missing from this graphic, and that is the panel of the people working together to take down that fence. So this is my challenge to you. I'm going to share with you the various activities that we've been doing. I want you to think about whether these are giving people boxes, taking down fences, or both. We've been working on this for a long time. Aoife McLysett and Sudhir Kumar started the Carer Awards, which was a very revolutionary idea to facilitate caregiver participation in our meetings. This could mean women with babies and other children. It could also mean single parents. It could be male parents. It could be people taking care of an aging parent. It could be people taking care of a disabled sibling or adult child. It could be anything you want it to be. And what was really exciting is that Aoife suggested that we could allow people to decide how to use their money best. So they say, I need this much money because I want to pay to bring my child with me to the meeting. I want this much money because I want to hire a caregiver to take care of my aging parent while I'm not there to do it. It could be anything. This was really exciting. We also have had undergraduate diversity mentoring awards, free on-site childcare at all of our in-person meetings, and paid charge waivers for our journals. I also want to tell you about some of our new initiatives. So this year we have meeting registration waivers. We have internet access awards to facilitate participation by people who don't regularly have access to high-speed internet. We've included mentoring and service as criteria into our faculty awards. We've passed a statement of values and a code of conduct for all of our society activities. And finally, we've set aside $25,000 every year for SMBE Idea Initiative proposals from the members. That's you. The goals are to increase representation and inclusion in our membership along all axes of diversity, and to collect data on and/or address systemic racism, sexism, colonialism, and any other exclusion that's going on in our society. If you want to help, we want you. We're recruiting members for the SMBE Idea Task Force, as well as proposals for the SMBE Idea Initiative. You can contact Dr. Lulu Stater at smbe.contact@gmail.com for more information. I'd like to also acknowledge all of the people who helped me get here. Mentors past and present, which would include all the members of my lab. You've taught me at least as much as I have taught you, as well as all the other many people who have supported me throughout my SMBE journey and my scientific career. I also want to acknowledge you and thank you for listening to this talk. I look forward to comments and feedback either in one of the question sessions or via email. Thank you again.